This is the key.
When we want to move a skeletal muscle, the brain sends a message in the form of an electrical signal down the axon of a neuron that is close to a muscle cell. This electrical signal is called an action potential and travels all the way down the axon of the neuron until it reaches the synaptic knob. Once at the synaptic knob, the calcium channels open and calcium from outside the cell floods into the axon terminal. The flood of calcium causes the synaptic vesicles in the terminal to be pushed down towards the synaptic knob. Once at the knob, acetylcholine that was inside the vesicles gets expelled from the cell by way of exocytosis and dumped into the synaptic cleft. Once in the synaptic cleft, the acetylcholine travels towards the motor end plate of the skeletal muscle cell and attaches to ACH receptors, creating a chemical signal that opens sodium channels. As sodium ions from the synaptic cleft enter the muscle cell, a depolarization happens, and this initiates an action potential that travels down the sarcolemma. The action potential travels into T-tubules located on the sarcolemma and goes down the tubules and into the terminal cisternae of the sarcoplasmic reticulum. This action potential triggers a release of calcium that had been sequestered in the terminal cisternae by calsequestrin molecules. As the calcium rushes into the sarcoplasm of the muscle cell, it floods over the thin filaments that contain actin, troponin, and tropomyosin. The calcium then binds to troponin and causes tropomyosin to be wrenched off the active site located on the actin molecule. Once the active site on the thin filament is exposed, the thick filament that is composed of myosin tails and heads becomes attracted to the active site. It leaps towards the active site on the thin filament and binds to it, forming a cross bridge, resulting in the myosin head landing in a low energy state. The sliding action of the two filaments and forming of cross bridges from the thick to the thin filaments repeats and pulls to contract the muscle. An ATP molecule then attaches to the myosin head. The energy released from the ATP molecule not only detaches it from the thin filament, but it also rotates the head back into a high energy configuration awaiting the next muscle contraction. ATP is then converted into ADP. The calcium in the sarcoplasma is pumped back into the terminal cisternae by active transport, and the tropomyosin covers once again the active site on the actin, relaxing the muscle cell. The end.
This is a vesicle, a bundle of muscle or nerve fibers ensheathed in connective tissue. Multiple fascicles bound together constitute a muscle or nerve as a whole. This is a sarcomere. In skeletal and cardiac muscle, the portion of a myofibril from one Z-disc to the next, constituting one contractile unit.